Very good afternoon to all. Myself Dr. Balwinder Singh from Department of Biotechnology, Khalsa College, Amritsar. I am here for short lecture on topic in vitro production of beneficial bioactive compounds from plants. Basically in plants there are two types of plant metabolites, that is primary metabolites and secondary metabolites. Primary metabolites are involved in growth, development and differentiation of plant. They are essential for the normal growth of the plant continuously synthesize these primary metabolites during its growth phase. Primary metabolites participate in various metabolic processes like respiration and photosynthesis. Primary metabolites are almost identical in most of the plant species. For example, all plant species synthesize sugars, amino acids, tricarboxylic acids. Proteins, nucleic acids, and polysaccharides are another examples of primary metabolites. Primary metabolites are basically building blocks and energy sources in plants. When we can consume different plant parts, we also get nourishment due to primary metabolites, and plant also get nutrition due to primary metabolites. On the other hand, there are secondary metabolites also produced in plants. Scandi metabolites are not involved in growth, development, and differentiation of the plant. They basically originate from primary metabolites as byproducts. They are important for several functions such as protection, competition, and species interaction. They are not essential to sustain the life of a plant, and they are not continuously produced in plants. They are produced during non-growth phase of the plant. In fact, we can say Scandi metabolites are the end products of the primary metabolites. The examples of Scandi metabolites includes alkaloids, steroids, phenolics, essential oils, lignins, terpenoids, waxes, flavoring agent, fragrances, etc. They are produced in very low quantity in plant and at specific portion or specific site of the plant. They basically participate in defense reaction of the plant. For example, in plants they provide protection against microorganisms like bacteria, viruses, fungi and protozoans. They also protect plants against herbivores like insects, worms, and animals. They also attract pollinators like bat, birds, and insects. So these Scandi metabolites are basically have protective function in plants. They are very useful for human beings because they have medicinal value. They have health benefits. So that's why we are interested in production of Scandi metabolites in plant tissue culture laboratory. These Scandi metabolites are also called as bioactive compounds because they have biological activity or bioactivity. For example, they have antioxidant activity, anti-inflammatory activity, anti-cancer activity, antimicrobial activity. So due to these different types of activities, they are important for current and future use in medicine due to their pharmacological properties. I am giving you very good example of Scandi metabolites. There are two Scandi metabolites, vincristine and vinblastine. These are the two important Scandi metabolites synthesized by Catharanthus roseus. These two metabolites have anti-cancer properties and they have very high market value. Market value of vincristine is around $2 million per kg, and market value of vinblastine is around $15 million per kg. So they have a very high market value. And they are commonly used in lymphoid cancer. They prevent polymerization of microtubules during division of the cancerous cells. So there are many such examples of Scandi metabolites which have high medicinal value. They are commonly used in different types of drugs in pharmaceutical industries. So that's why we are interested in producing these Scandi metabolites under in vitro condition in plant tissue culture laboratory. We can synthesize also them, but that process is very costly and it takes a lot of time. So these bioactive Scandi metabolites can be synthesized by taking the explant from the plant. Explant may be cell, tissue or organ. For example, we can take even leaf, root, sepals, petals, part of the fruit to produce Scandi metabolites. Now, why we should produce these Scandi metabolites under artificial condition in plant tissue culture laboratory? There are many advantages of producing these Scandi metabolites. The rate of cell growth is very high in plant tissue culture laboratory as compared to the plant cells growing under natural condition. Another advantage is that biosynthesis can be initiated from very small amount of explant. We do not require large portion of the plant to produce Scandi metabolite. We can produce Scandi metabolite from even small root segment, or even small sepal segment of the plant. So required material is very small. Another important advantage is that we can control the conditions of the cells growing under artificial condition, which is not possible under natural condition due to variable environmental conditions, due to change in climate, due to change in temperature, light. So it ensures continuous production of Scandi metabolites in tissue culture laboratory when we produce them under artificial condition in culture tubes. So it ensures continuous production and it is not dependent upon the environmental condition. It is independent because we are controlling everything inside the laboratory. Another advantage is that why we should produce Scandi metabolite in laboratory is that we can also incorporate precursors of Scandi metabolites in a culture medium. These precursors are used by the plant enzymes to convert into important products. So we get important products very easily when we incorporate important precursors of the Scandi metabolites. So plant tissue culture offers mechanism of incorporating precursors in the cell culture on the medium so that we can produce large quantity of Scandi metabolites. Another advantage is that we can also induce soma clonal variations. Soma clonal variations are basically variations in somatic cells during their repeated cell divisions. So sometimes these variations are useful and we get cell lines which are more efficient in producing Scandi metabolites. So we can use soma clonal variant cell lines which are more efficient in producing Scandi metabolites in tissue culture laboratory to produce large scale Scandi metabolites. Moreover, plant tissue culture method is economical as compared to natural method because we have to harvest the plant, we have to first grow, then harvest, then extract Scandi metabolites. So if we compare it with naturally grown plant tissue culture of method is more economical for the production of Scandi metabolites. So there are many advantages. However, some technical challenges are also there in Scandi metabolite production in plant tissue culture laboratory. Establishment of tissue culture system is difficult for many plant species. In woody plant species, we do not get any response. Plant cell does not divide. They do not develop cell suspension culture. So that is the another challenge, that most of the woody plant species does not show proper cell division, proper cell growth in cell suspension culture. So establishment of cell culture is difficult in some plant species. Then another technical challenge is to design a bioreactor system. We require specially designed vessels for the production of Scandi metabolites from the specific explant, from the specific free cell suspension cultures. So that is another technical challenges. Now there are many technologies for production of Scandi metabolites. The common technologies includes cell suspension culture, plant cell immobilization, plant cell elicitation, hairy root culture, and biotransformation. First of all, about free cell suspension culture or cell suspension culture, it is the most convenient method for production of Scandi metabolites. We select mother plant. Mother plant is the plant from which we take explant. We take explant from that plant, culture it on a medium form callus from that explant. Once the callus is formed, then we multiply that callus to get large number of undifferentiated cells. These undifferentiated cells are further transferred in a liquid medium to get free cell suspension culture. When the cells are present in the liquid medium under shaking condition, then we call them free cell suspension culture. Once we establish free cell suspension culture, that suspension culture is then transferred in a bioreactor. In bioreactor, we get continuous production of Scandi metabolites. After production of Scandi metabolites, we separate and purify the Scandi metabolites and supply these Scandi metabolites to pharmaceutical industries. So it is the most convenient method for large scale production of Scandi metabolites in tissue culture laboratories. It is a flexible system. It can be manipulated to increase the production of Scandi metabolites by changing the culture condition, by changing the light, temperature, humidity, etc. It is also very useful to transform the cheap and plentiful substances into expensive substances which have medicinal value. So, free cell suspension culture is the easiest method of producing Scandi metabolites. However, in this method, there is a problem like cell aggregation, shear sensitivity, oxygen transfer, genetic instability, low growth rate. So these problems can be overcome by second technology, that is plant cell immobilization. In plant cell immobilization, we entrap the plant cell in alginate matrix, or in polyurethane foam or polyester fibers. We entrap the cells, we manipulate the environment around the cell so that it can produce more amount of Scandi metabolites. It can overcome the technological barriers of shear sensitivity, cell aggregation, instability. So these problems can be overcome, which mostly we found in free cell suspension culture. The problem includes cell aggregation, shear sensitivity, genetic instability to cells. So now next technology is plant cell elicitation. In this case, we use elicitors to enhance or improve the biosynthesis of Scandi metabolites. We use different type of elicitors, that includes biotic elicitors, abiotic elicitors, and these elicitors are very effective to enhance the production of Scandi metabolites because they interact with the plant cell mechanism, metabolic process to enhance the production of Scandi metabolites. These elicitors may affect synthesis, rate of synthesis and accumulation of Scandi metabolites in plant cells. Elicitors are basically used to enhance the production of Scandi metabolites. But the exact mechanism by which elicitors works is not exactly understood. Next technology is biotransformation. In this technique, we use substrate. We provide substrate to the plant cell growing on an artificial medium so that it can convert the substrate into important product. So, in vitro plant cell and organ culture act as a biocatalyst to perform a complex reaction. It perform a complex reaction and convert the provided precursor into important Scandi metabolites. We have produced variety of compounds like steroids, alkaloids, terpenoids using this biotransformation mechanism. Next technology is hairy root culture technology. This technology is basically used to produce Scandi metabolites that are mostly synthesized in the roots of the plant. So we can say root associated Scandi metabolites can be synthesized using hairy root culture technique. In nature, there is a bacteria known as Agrobacterium rhizogenes, and it is responsible for causing hairy root disease in plants. And when we use this Agrobacterium along with plant cells to produce roots, the roots are capable of producing Scandi metabolites like that of natural roots. We take root as an explant, then we form callus, then we co-cultivate the callus with Agrobacterium rhizogenes. This results into genetic transformation and we get hairy roots. And these hairy roots have same properties like that of natural roots. They are capable of synthesizing Scandi metabolites. So hairy root culture technique is very useful for production of root associated Scandi metabolites. So these are different types of technologies that we can use to produce Scandi metabolites. Now at the last is limitations. Although there are many advantages of this technology, there are some limitations. First limitation is that scale up system is difficult. It is very difficult to scale up plant cell culture system using specially designed vessels or bioreactors. Another limitation is that plant cell size is larger and it has more physiological requirements as compared to microbial cells. Microbial cells can easily be established in bioreactor, whereas plant cell culture system establishment in bioreactor is difficult. Another limitation is transfer of oxygen. Oxygen transfer is a problem in plant cell culture because plant cells have tendency to aggregate. So it is difficult to provide oxygen to cell aggregates. Another limitation is that some of the Scandi metabolites are not secreted out of the cell. They are retained inside the cell. So when they are retained inside the cell, then we are facing difficulty in harvesting such type of Scandi metabolites. Another and last limitation is that recycling of cell culture is difficult. So due to non-recycling process, it increases the cost of Scandi metabolite production. So thank you very much. Thank you for listening.